For more, Boris is going to explain the second half to get into the real math of the talk. I don't know.
It sounds like that. Okay, so I think I got the general philosophy out of the system, and let's actually try to understand something about the rate of adaptation. So first, addressing Oscar's example of adaptive mutations in asexual population. Okay, so we're talking about population with fitness diversity, so let's refer to this fitness axis, let's label it X, and generally refer to fitness differential relative to the mean in the population. And now we have our little bin distribution, and very often we want to describe this simply by continuous function approximation. We'll take this continuum limit, and the simplest thing is just to write down a little mean fieldish type equation for dynamics of this distribution. So clearly individuals that are better than mean in fitness grow exponentially with a rate given by this differential, and this is a very simple little equation. Basically tells us that the number of individuals above the mean is going to go up, the number below the mean is going to go down, and the whole thing is going to translate to the right with some velocity, where this velocity is basically the rate of increase, in this case, of the mean fitness. And this is simple enough. You easily solve this and demonstrate that the solution is simply the Gaussian with the mean translating forward with a constant speed equal to the variance in this distribution. And the rate of adaptation proportional to the variance is Fisher's fundamental theorem. So the only thing is the Fisher fundamental theorem is correct. The only thing is it actually doesn't tell us anything about how much variance there is in this distribution. So in fact, it's not just one solution. So we have this traveling wave solution, but it's not just one solution. It's a whole family of solutions which have different width and move with different speeds. And of course, this can't really be true in any finite population, because with what we have here right now, in the finite population, even if we model this by continuous distribution, we know that we have sampled it only to a given depth, and there is maximum fitness in any finite population, right? So for a Gaussian, we know, for a Gaussian with n samples, we know exactly what the expected value for this maximal fitness is in the sample, and it is only logarithmically or square root of a logarithmically dependent on the population size. And there'll be lots of logarithms of population size in this talk, and pretty much all of them are in one or the other way related to this fact. So relation between so the width of a distribution and its leading edge. So, okay, why is this important? Right? This Fisher traveling wave moves forward because so the imaginary individual way ahead of the front will eventually get a chance to get amplified and take over the whole population. But if there isn't such an individual, there is no mechanism of producing it, right? So in a finite population, this Fisher wave will basically slam into its leading edge, the variance will collapse, and it will stop. So to really calculate what the variance in the distribution is and what the velocity is, we have to so start thinking about how the leading edge here is going to move forward. And there are two effects which are going to do this. There is some probability of a mutation occurring somewhere, some individual in this distribution, and advancing it from point x prime to x. So this is just a parameterization of mutation effect distribution, right? So general that it virtually says nothing. And the second effect, which we will talk about in a moment, is recombination. So actually in the sexual population, right, two individuals from the population will come together and make a baby at the fitness, with a fitness related to ancestral fitness, but not the same.
Well, you said these are so general they say almost nothing, but it's not quite, right? I mean, they say that these kernels only depend on the individual's fitness.
Yeah, so, you know, yeah, this definitely says something. So, okay, so let's talk about, Oscar's model here, which was worked out by Michael Desai and Daniel Fisher. Daniel, I'm told, was here yesterday. Well, anyway, so let's, for simplicity, say that they're only beneficial mutations and they all have characteristic size. So that basically means that the mutation falling in a given bin can move the offspring one bin to the right. And so the new fitness territory will basically be colonized by mutated offspring of the nose of individuals in the nose of this distribution, in the top bin, that maximal fitness again, right? And in order to calculate the rate of adaptation, well, what we have to do is the following. Mutation rate times population size, that's the rate of mutational influx into the population. Each mutation carries benefit S. So, but then the all-important factor is what is the probability of fixation? Which of these mutations that are occurring everywhere in this distribution have a chance of fixating and taking over? And the insight of these two gentlemen was to realize that it's basically just the top bin mutations that mutations falling into the top bin that have a chance to fixate, and the offspring of the vast majority of this population are basically never going to make it. So genotypes, typical genotype is basically doomed for extinction, right? And it's the rare genotypes in the nose of the distribution that are going to take over the future. So very crudely, what I just said is a statement that only the fraction of all mutations, the ones that fall into this top bin, right, number of individuals in the nose over the total population, right, that's the fraction. Only they will have a chance at passing on their genes. And then in addition, not all of those either, since we talked about stochastic extinction and survival probability in the face of the drift, which we said was proportional to the fitness advantage. That's the difference between the nose here and the mean. And now you observe that this big N factor actually drops out. The only thing that's important is how big is the nose, and the whole wave is moving forward, and the stop bin exists only transiently. right? Until the new mutation comes in and seeds yet another bin, right? And so it is growing exponentially for a time such that mutation rate times the size of this bin is equal to 1. So anyway, I don't think I really want to walk you through the arithmetic here, but all I want to impress you with is the notion that it's a calculation, but it's not a terribly deep and profound one, and you can do it at home, and especially with Desai-Fisher paper in hand, and it just illustrates this very simple idea that the most important thing in understanding the rate of adaptation is to understand the probability of survival for the new allele. And that very much expresses the underlying dynamics of clonal competition. There are all these many clones that are generated by mutations in different backgrounds, and they're all competing. But only a small fraction of them are going to survive. And now, if you actually work out what this arithmetic tells you, you will find, okay, with my simplified arithmetic here, so I think there are some experts in the audience who are going to immediately correct me that this is not quite the right result. But all I want to show you here is that a linear dependence is replaced by a logarithmic, much weaker dependence on the population size. And the real expression you'll find in Desai-Fisher. But the guts of the calculation we captured in just 3 lines on the previous slide. Okay, so I obviously plan to do this in my first lecture. And now we're going to start talking about the role of recombination. And the importance of interactions. So we just talked about one particular kind of an interaction. So that was an interaction between different clones in an asexual population as they compete against each other. So suppose this is a population, and this bunch of beads is a genotype, and in red is genetic variants, and there could be many individuals that are genetically identical, especially right in an asexual population that will happen. So these form a clone, and then different clones, this clone A competes against clone B. So we've just discussed this in great detail. But there are other important interactions here. So there are many alleles that contribute to the fitness of the clone. And you may be interested, as we're just talking about the probability of fixation for a given mutant allele, right? So the probability of fixation for a given allele depends on which genotype it lives on, right? So, and this allele alpha, for example, sometimes will be coupled to the fate of allele beta. Sometimes it will be coupled to the fate of some other allele. And even a deleterious mutation can rise to prominence in the population if it is riding on the back of beneficial mutations. So that's called genetic hitchhiking, so otherwise known as genetic draft. So we were talking before about genetic drift. So this is just sort of non-heritable stochastic variation in offspring survival, right? But this hitchhiking contributes another element of stochasticity into propagation of the allele. Right? So now it depends on which background this allele arised on. And once we turn on recombination, of course, this allele will be able to hop from one background to another. So the effect of genomic background on the propagation of that allele, from the point of view of the allele, will look like sort of a stochastic force. So sometimes sort of a good background will help the spreading of the allele, bad background will hurt the spreading of the allele, and in fact, in a genetically diverse population, the effect of the so-called genetic draft dominates the stochastic effect of drift. So that's another kind of interaction we want to talk about. And the third one, of course, is the very fact that phenotypic contribution of any pair of alleles is not necessarily equal to the sum of their individual contributions, right? So that's known straight as epistasis. And
Boris?
yeah.
Real quick. Why do you separate them into hitchhiking and clonal interference? It seems to me like hitchhiking is an example of clonal interference. It might make more sense to separate them into interference because they occur on the same background and interference because they occur on different backgrounds and compete with each other. But it doesn't really matter if they're beneficial or neutral.
I don't really want to enter a debate over my use of words on my time. So let's do this after this clock stops ticking. Okay. Okay, the effect of these interactions strongly depends on the rate of recombination, right? So, for example, clearly clonal interference, competition between different clones, or at least the outcome of it, strongly depends on so the possibility of recombination to bring these two different alleles, put them on the same genotype, and perhaps build a better genotype if those two were beneficial, right? On the other hand, this effect of hitchhiking or background selection, so the fact that the deleterious allele may be hiding behind the beneficial allele sitting on the same genome, is also ameliorated by recombination, right? Because a recombination event will set these two alleles asunder, and now all the deleterious effects of our allele beta will be seen by natural selection, and recombination therefore will increase, if you like, the effectiveness of natural selection acting on individual alleles. So this is sort of a distinction between natural selection acting on genotypes, right? And in an asexual population, that's sort of all there is. There is the whole genome of an individual, right? And the sexually reproducing population, which is really, at least in some limit, can be thought of as a soup of alleles that are being sort of continuously reshuffled and transiently put together on just one genome and one individual, and if you like, get tested by natural selection in one particular combinatorial reassortment. So let's first think about what sex and recombination will do to the rate of adaptation in this large, rapidly mutating populations where selection is confounded by clonal competition. Okay, so suppose the new mutation comes in and falls, happens to be an individual which is much better than the mean in the population. Well, so carriers of that mutation are going to expand, are going to proliferate in the population. So now I guess my time is going this way, and the width here represents a fraction of the population carrying that new mutant allele. So it is rising. The only problem is that mutation rate is high, population is large, so other genomes are also getting beneficial mutations. Everybody is getting better, and now suddenly our particular clone carrying a given mutation finds itself worse than average and now starts shrinking. Right? At this stage, it's basically doomed for extinction, except perhaps with a little bit of luck. One of the offspring recombines with a better genome and transfers this mutant allele that we're tracking onto a better background. Right? And now this process starts again. And so now basically think of a given allele surfing on this evolutionary traveling wave. And the trick is for the allele to maintain presence at the leading edge long enough so that it can actually expand and fixate in the whole population.
Why do you ignore the sort of surfing effect due to mutations, not due to recombination? So just having a mutation that brings you up to the next higher fitness class.
Yeah. No, I don't want to ignore anything, right? So if you like, you can recast this whole Desai-Fisher story exactly in the same language. And in fact, the calculation is going to be pretty much the same. I mean, the arithmetic is a little different. The result is a little different. But logic is very much the same. So let's go over this again. So we got the population, we got a new mutation, starting a clone on the background X, and then many of its offspring are going to recombine onto different backgrounds, and some of them will be worse off than they started on, and some of them will be better off. And one way I can redraw this picture is sort of not even try to represent the size of the clone, sort of number of individuals with that genome, but just keep track of their fitness. Right? So each little bubble here will turn into a stick, and time goes this way. So this is where this clone first came into existence, and that's where it got extinct. Or I guess this was our first clone. And before it got extinct, it seeded a few new clones. And then those clones seeded a few new clones, and so on and so forth. And this, of course, is happening alongside with this adaptive shift in the whole population, which we know how to describe as a Fisher traveling wave. Of course, this Fisher traveling wave is sort of very mean field description, which really pays no heed to fluctuations, whereas my picture here is really trying to zoom in on the fluctuations. And but the bottom line is these two pictures have to agree with each other, because this population ultimately is just a superposition of all the little clones that are jumping back and forth, right? So what we need is a way of arriving at self-consistent description of the dynamics in terms of this very stochastic process involving individual clones, and so the mean field description of the whole population in terms of Fisher's traveling wave. And it is exactly this effort to self-consist the two pictures that will fix the variance in a distribution, right? That fundamental theorem of Fisher by itself cannot do for us. Let me redraw this picture in terms of a numerical simulation, which you can easily do, right? And so still the same sort of stick diagram, and these little sticks are individual clones, and every so often you see a new clone way ahead of the rest of the population at that time appears, and actually the color coded here is the size of the clone. So it grows very large, it has lots of offspring, they colonize many genetic backgrounds and prosper for a while, but eventually nevertheless go extinct, right? And then every so often, so there is some probability that in this process, the offspring of a given allele will colonize the whole distribution, right? We'll go to fixation. Now our job is actually to calculate the this non-extinction probability, right? The probability to succeed rather than fail. And then we're going to use exactly the same general philosophy. The total mutational flux into the system times the survival probability will determine the rate of evolution, rate of adaptation, and the width of the distribution. So now let's actually try to calculate. So we have this little bubble, and the first task, while there are very few copies of this allele, for this allele is not to become extinct, right? Let's calculate the extinction probability, right? So with probability almost 1, it will become extinct right away, and that's the end of the story. But with the probability 1 minus this extinction probability, which is to say x over 1 plus x, it will not become extinct right away and will grow into a largish clone. And then it will have an opportunity to recombine and start new clones on different backgrounds. So let's count. What's the probability of having little m offspring? And you can convince yourself that it's some Poisson process. And right now I'm just summing over all possible outcomes. So this obviously is 1. And then this is 1. So this is just one complicated way of writing 1 is equal to 1. So nothing quite happened yet. But we haven't really calculated what we wanted to calculate, which was the extinction probability, right? So for the offspring, for this allele to become extinct, now all of its offspring have to become extinct, right? So now we take these m offspring, they live now in different backgrounds, and we say that the probability of extinction of the whole thing is just given by this expression. All of the offspring have to become extinct. So now we got ourselves an equation. It depends, of course, here on where exactly the new clones, what new backgrounds they have fallen on, and we have to average over that. And we'll have some model of recombination to tell us where the recombinant offspring are going to be distributed in the distribution. And now we got ourselves actually a well-defined equation that allows us to calculate the extinction probability. And the only thing that I need to add to this is I defined as gamma, basically the size of this clonal bubble as the clone first grows exponentially, especially if it is fitter than average, that is to say X positive. But at longer times, when the distribution catches up with it, it collapses. So the size of this clone is very strongly dependent on the background, right? It's e to the x squared, right? So that's actually why the leading edge of distribution is so important. It all traces to this fact that mutations that fall on the backgrounds that are much better than average are going to rise to larger copy numbers, will have a higher chance to spread over many, many other genetic backgrounds, right? Generate many offsprings, and therefore, if you like, hedge bets, right? Not all the eggs are carried in the same basket. So one just proceeds with this calculation, finds the survival probability, and for the reasons that I just explained, the survival probability increases so drastically as one moves towards the front of the distribution, and in this way, we have succeeded in defining this probability of non-extinction, the probability of survival. So we already talked about this very general expression. And now this has turned into an equation that we have to, well, of course, the survival probability depends on how broad the distribution is, right? We have to calculate it as a function of the breadth of the distribution, right? So sigma squared, the variance of a distribution, when we're calculating survival probability, is just a parameter, right? But now, thanks to Fisher's theorem, we have a relation, right? So the velocity, speed of adaptation is equal to the variance. Now we have an equation that actually allows us to determine that variance that we didn't know to begin with. And the result is as follows. For large mutation rates, large population sizes, we still have only logarithmic rate of, the speed of adaptation still only depends on the logarithm of the population size. So in that sense, sex and recombination have failed to completely eliminate clonal competition. But the prefactor of that logarithm does increase with the frequency of recombination. And in fact, what one sees is this benefit of sex manifests itself in greater rate of adaptation for higher frequency of recombination, right? Even though still a lot of mutations are wasted. And they're wasted, again, you see it right here. The survival probability is not 1 through the whole distribution. It's still only a fraction that survive.
So before in the asexual case, even when the rate of adaptation became only logarithmically dependent on the population size and the mutation rate, you could still crank it up by increasing the strength of selection. And here, it's actually just recombinations. S doesn't even matter that much.
Well, S matters still because it defines sort of the crossover between the two regimes. So,
Boris,
yeah.
this says that you adapt faster if you just keep increasing the recombination rate.
Mhm.
What's the intuition behind, or do you have a reason why recombination rates aren't just really high then?
Yeah. So, needless to say that this is only half, epsilon, of the story. So, I'm talking about high mutation rate here, but strictly speaking, only talking about relatively low mutation rates. So, compared to the width of the distribution. In the following sense, so strictly speaking, you're thinking of a facultative recombinator, right? Something like a virus, which doesn't really have to recombine to reproduce, but will sometimes. Or like a plant, right? Which will outcross sometimes, but not always. So now, really, we're thinking of relatively low recombination rate, and your question is completely valid. So, having said that, why isn't it arbitrary high, right? So, good question. One of the things that immediately comes to mind is another kind of an interaction, right? Which is epistasis. Right? So here we're talking about recombination bringing beneficial alleles together onto the same genome, right? But the assumption is that everything's additive. Now, suppose you have genetic interactions where synergistic genetic interactions, right? So then you'll have a cost of recombination because you'll start breaking them up, right? So there is something known as outcrossing depression or outbreeding depression, right? So if you have two even sexually propagating populations that are reproductively, well, I shouldn't say reproductively, geographically isolated, basically fungi growing on the different stones, right? And you bring them together, cross them, and you look at the fitness of the offspring. It is systematically lower than fitness of both of the parents, right? And the explanation is that the two populations have fixated, or at least have large fractions of pairs of alleles which work well with each other, but not so well if you mix and match between two different populations, right? So things like this immediately kick in. So basically, and you can think of this as a sort of a crossover between selecting on genotypes or the genomes versus selecting on the alleles, right? So, right, if we push this rate of recombination sort of arbitrarily high, or for that matter, just make a genome an assortment of tiny little chromosomes, which are continuously getting reshuffled, right? You'll be selecting only on alleles, right? So you will never allow these sort of epistatic interactions come in and establish. So that's sort of, I guess, one way of, yeah, please.
I have a question about the other extreme. So as the recombination rate goes to zero, it seems that this result does not converge to the Psi-Fisher result. Could you comment on that?
Yeah. So, so it's a subtlety. It's a rather interesting and complicated subtlety. So having to do with sort of the crossover between Now this curve and a similar version of this curve that you would calculate for Desai-Fisher. Technical and subtle issue. So mathematically, we cannot push this calculation through the limit of rate of recombination smaller than benefit of a single allele. It's a technical little thing. Okay. Going wrong way. So how much time do I have?
15 minutes. Wait, no. No minutes.
Yeah.
No minutes. I'm sorry. You're out.
15 minutes.
I was confused. Uh-oh.
Okay.
Yeah, so I guess you got to wrap it up.
Okay. It's fine. It's modular. I was going to tell you how, having done this much of a calculation, one can actually push forward and understand a little bit about draft, which is that other kind of genetic interaction, which somebody here said, well, was not so different.
Joel, I think.
And indeed it isn't. The calculations are really very closely related. So when we worry about draft, we're talking about propagation of a given allele as it hops from one background to another. And I was just waxing poetic on that very subject for 15 minutes. And it's just that in order to describe draft, we want to actually calculate the probability of so a given allele to rise in number to any given n, as opposed to just the probability of it going extinct. So we can do that calculation. And the outcome is that you learn something that population geneticists, some of the population geneticists have realized before, that draft is really quite different from drift. I'm sort of realizing that given that I have 0 minutes, maybe I shouldn't really attempt to go into any detail, except say that there is a rather widespread use of this notion of effective population size. So the effective population size is this notion that if you have this draft in the population, it introduces the additional stochastic, if you like, force into dynamics of alleles, which can nevertheless be captured simply by saying that the population, so effective population size, is smaller than the census size, the number of individuals in the population, right? And this effective, so smaller population size, description of, and the neutral model with the small effective population size will then capture the dynamics of the population with fitness diversity, where alleles hitchhike and sort of boost and bust each other. And the bottom line is that's not so. And that actually has to do with the statement that I made in reviewing the coalescent and the statistics of genealogical trees, right? And I said that fitness diverse population has a different statistical structure of the coalescent, right? So if the effective population size description was valid, it wouldn't be so. It's just the height of the tree would get rescaled, but the statistical structure inside would be the same. And it's not so, and you can actually calculate various things about it and convince yourself, or rather learn, just exactly how not so it is. And I was going to tell you a little more about this, and I won't, and I won't, and I'll just conclude. So where do we stand? So, in the last 10, 20 years, there's definitely been progress in understanding dynamics of natural selection in these rapidly evolving, fitness diverse populations. But we're still a little bit in the state of this proverbial bunch of blind natural scientists exploring, feeling up the elephant from different sides and sort of disagreeing about their experience from touching the tail or the trunk there, right? So we have, and I think that was sort of very visible in Oscar's talk, because he presented a little model and immediately there were lots of questions. But how is this conclusion dependent on the details of that particular model? And surely that model is not a reality, and therefore it's irrelevant. And then there are all these different models. But what we need is some sort of a grand unification of these models. And we're already beginning to realize that many of these different models studying different aspects of the phenomena, be it Mueller's ratchet or adaptation, actually have a lot in common. And I think the challenge for us is sort of to continue weeding model details away from the general phenomena. And this may sound to you like another lot of words, but I think there is a reasonably well-defined substance in it. And it goes back to our discussion of statistical properties of the coalescent. What we're seeing is the statistical properties of the genealogies have aspects in common which completely transcend the details of the model. So the genealogy that you will see in a population with only deleterious mutations and a purifying selection, right? So that will set up some mutation selection balance and fitness diverse distribution. And you can ask, what are the statistical properties of the coalescent under these conditions? So these properties are very similar to the properties that you will see in a continuously adapting population, which just moves forward as a traveling wave. And if you say that they're both beneficial mutations and deleterious mutations, then you can have a fitness diverse population just sitting somewhere in the fitness space and just fluctuating a little bit either in yuan, and it will still have essentially the same statistical properties of the coalescent. So the details of these models, therefore, we can hope, are not terribly important. And what we need is to define, so back to statistical physics, to define some equivalent of so thermodynamic variables and order parameters, some empirically measurable quantities that will parameterize the relevant structure of the population, quite independent of the details of natural selection, et cetera, which are setting up the distribution in the first place, right? Because this way we can actually hope to make progress just by looking at observations. For example, just observe the genealogy of a population, right? And one can do this by some extensive sampling and analysis. Let's look at that, figure out what the relevant parameters are. We're not going to learn everything about every bloody polymorphisms under selection, but perhaps we can learn something which is even more useful in practice, such as the ability to sort of forecast near future. So I'm finished with this, again, philosophical flourish. And pretty much everything that I talked about involved a collaboration with Richard Nair. Well, actually, there were lots of things that I talked about which didn't involve me at all. And some of those still involved Richard. So Richard used to be at KATP. Unfortunately, he has moved on. Well, I guess that's a natural course of events. So he is in Max Planck in Tübingen. Sid Goyal is another postdoc at KATP. He's actually about to move on and to Toronto, and unfortunately I didn't really get to talk about what we've been doing together. And Adel Dayarian is another postdoc at KATP, and together we have this little paper on actually trying to predict the future based on genealogies, but I had to skip that part of the story. And Daniel was here yesterday, and I'm sure you've seen him in action, just like in this picture. So we really enjoy working together on occasion. So thank you very much.
Squeeze in the question.
So you mentioned recombination as being this trade-off between taking advantage of beneficial mutations and epistasis. So if we take the empirically observed rate of beneficial mutations and the recombination rates, and assume that the recombination rate is somehow optimizing for this trade-off, so can we actually get some estimation of the amount of epistasis in various genomes?
Well, so I would have actually very much liked to do something like that, right? So I'm pretty sure that, well, actually, I'm not sure. I know that you can certainly do this in a model. So we can write down some reasonably well-defined model which will have things that you just specified. We'll parameterize so fitness landscape by some suitably defined epistatic interaction term, and we can make your favorite assumption about frequency of beneficial mutations and do a calculation and arrive at some optimal. But I think you want to do more, right? You want to actually try to prove based on existing data that that is the case. Well, I'm not sure we know enough that we could actually falsify or prove this statement, right? And I think the fundamental reason is the statistical physics attitude is good for explaining functional relations, right? And not so good at actually predicting numbers, right? So it is much more plausible that we can get sort of a correct prediction about, say, dependence of a rate of adaptation on population size. And perhaps, the prefactor of a logarithm can be, a fitting parameter there, right? But we can still test the functional dependence, right? Where it comes to trying to predict that the frequency of outcrossing of Saccharomyces cerevisiae should be X, I'm a little more skeptical, just because it actually requires sort of very, very specific, precise knowledge of various parameters. Having said that, I think if you were to try to do this in a lab experimental setting, where you have a better chance of controlling selective pressures on the yeast, then maybe one can try to do it. Right? And there are sort of various bits of evidence suggesting that the frequency of outcrossing is under selective pressure. For example, C. elegans, right? Mostly selves, but outcrosses with some small probability, and that actually can very easily vary from 1% to 50%. And I know that people are sort of trying to study how it depends on sort of different selective regimes, and for that matter, on sort of presence of genetic diversity in the population. And I think the evidence is that it actually is surprisingly readily readjusted. Right? This is a long answer to a short question.
Yeah, actually, we've already used up more than half our break here,
Yeah.
so I think it's time to thank Boris.